Hello, and a very warm welcome to everyone who has joined us today. My name is John Norman, and I'm the moderator for today's webinar, in which we will hear about the power of artificial intelligence when applied to agrochemical bioactivity. The group will be discussing the challenges of growing agrochemical resistance and increasingly stringent regulatory requirements. They will draw on case study evidence to examine how deep learning techniques are enabling us to address and overcome many of these challenges. So let's get things underway, and I'll pass things over to our host and presenters, who once again are George Lahm, Laurie Christianson, Bailey Montefiore, and Matt Segall. Matt, I'll now hand things over to you.
Thank you very much, John, for introducing today's webinar. It's a great pleasure to see so many people in the audience, and particularly a great pleasure to welcome our guests, George and Laurie. Just before we get started, I'll just say a few words of introduction to our guests. George is a distinguished fellow at FMC. He's an organic chemist with a 40-year career in agrochemical research. He's been a leader and inventor in the discovery of a variety of new products for insect control, including the sodium channel blocker indoxacarb, the ryanodine receptor activators Rynaxypyr and Cyazypyr, and the nematicide fluaza-indolizine. I knew I'd make a mess up with that. These products are landmarks in the protection of food crops and demonstrate outstanding pest control with high environmental and non-target safety. George has been recognized with numerous awards, including the Agro Lifetime Achievement Award and the American Chemical Society's International Award for Research in Agrochemicals. Laurie is a senior R&D scientist with FMC. She received her PhD from the University of Wisconsin-Madison. Prior to FMC, she was a computational chemist and scientific data analyst at DuPont CR&D in crop protection. Her research interests include ligand-based data modeling and design, applications of machine learning to compound screening, and quantum chemistry applications in agrochemical research. And last but not least is our main speaker today, Bailey Montefiore. Bailey is a senior scientist here at Optibrium. She did her PhD at Newcastle University in the field of structural and molecular biology and a postdoc at Imperial College London before joining us here. She spent her first years at Optibrium on the application science team and later joined the research team, where her primary focus is the application of deep learning imputation to projects such as the one she's going to present today. Before we get started with the main presentation, I'd just like to say to George and Laurie, thank you very much for joining us for today's webinar. It has been an absolute pleasure to collaborate with you and your colleagues to explore how deep learning techniques can be applied to agrochemical discovery. And before we get started, I wonder if you could share a few thoughts with the audience on the challenges that the industry is facing and what drew you to explore a collaboration with us at Optibrium in the first place.
As far as, I think perhaps George could speak more to the challenges of the industry, but as far as why we became interested in the first place, I have worked with Stardrop and Optibrium products for, probably the better part of a decade now. And as you started to publish on Alchemite and on your machine learning methods, I guess I became very interested in what you were doing, and specifically some of the fit that I saw with your methods and our data. Because of the nature of agrochemical research, a lot of our screening is whole organism screening, and so we often don't have, for instance, the IC50s or just the data that a lot of methods expect to be interacting with by default. And so some of your presentations really looked like the method was going to much more easily be able to take in the type of data that we routinely work with and not have to require a lot of manipulation. And happily, I would say that's been borne out in practice. This has been a really easy experience getting the data into a form that Bailey is able to work with. I don't know if you want to add anything, George?
I can add a few things. So I'm a chemist, as Matt mentioned in the beginning, and really my interest over the course of many years now, 4 decades, over 40 years, has been finding molecules that control pests and solve the problems of regulatory requirements, pest management, pest resistance, and other things that were mentioned by John in the introduction. But all of that for us that work in crop protection by chemistry comes down to finding compounds that are essentially leads for new chemistry, new chemical areas that can lead us into you know, compounds that may work by a new biochemical mechanism. And we have a whole host of tests in place to help us understand that once we find these new leads. But the problem of finding the leads is actually the problem when you're right at the beginning. Now there are, with synthesis on demand and purchasable chemistry, there are billions of compounds available. You know, I really grew up in an environment where every idea you wanted to test, you actually went into the laboratory and made it, made the compound and tested the idea. And there's something to be said for that, and we would never want to give that up, because that very targeted research can't be easily done through the billions of available, even though that seems like a large number of compounds. But the second problem that creates is that we can screen a large number of compounds if we're just going out and looking for chemistry that is perhaps interesting. We can screen somewhere in the vicinity of 50,000 compounds a year in our high throughput screens to get an idea of whether or not we've got any associated biological activity. But 50,000 is really a drop in the bucket compared to multi-billions. You know, we could screen this for, you know, for thousands of years probably and not run out of chemistry. So we do need to be selective in what we purchase, what we make, particularly when we're looking at our high throughput screens. And that's always been difficult, and that's been something Laurie has really been addressing for the better part of the last probably 20 years. But what I really liked here is that we have this enormous volume of data and chemistry, and we have a link between the two because we've tested all of our compounds that are in our files in our screens. And in so doing, we've accumulated a really vast knowledge of what types of chemistry give us associated biological activity. And I guess I was really attracted to the idea that through this imputation, where you could take large volumes of data and large volumes of chemistry and somehow be predictive about what new types of chemistry might give you some biological response was very appealing. And the idea is not to actually find the compound. You know, when we're screening 50,000 compounds a year, the real goal is to just maximize the probability that you're going to be finding active chemistry. So if you raise that number from 1 or 2% actives across that number of compounds to 5 or 6%, that would be an enormous gain and would give us a lot more to work with. And then once we find the compounds that are interesting, then we deal with the problems of resistance and identifying mode of action. And so it's all connected, but it really starts out with we need the chemistry to work with. And so the ability to do that through AI, you know, it's really interesting for me to see this happening sort of towards the end of my career. But the ability to think about things this way is really interesting to me. And so I've also, like Laurie, found this project to be extraordinarily interesting, and I think you'll see that from Bailey.
Thank you so much, George and Laurie, for that background. And I'm now going to hand over to Bailey, who is going to talk to us about the application of deep learning imputation to prediction of herbicide and insecticide compound properties. So over to you, Bailey.
Great. Thank you, Matt, for the introduction, and thanks, George and Laurie, to you for setting the scene for what I'm going to talk about. So here's an overview of today's session. So first, I'm going to give a short introduction to agrochemical discovery in general and the data that we'll be working with, that we worked with in the project. And then I'll talk about the Alchemite method for deep learning imputation, including how we validated the models that were built. Then I'll go into the results. So I'll talk about the results against the independent test set. And then I'll talk about analysis of the uncertainty and how we can use the uncertainties that Alchemite calculates with the predictions in order to guide our decisions. And I'll go on to virtual compound prediction, how we can use the predictions to select the most promising compounds, and also talking about importance analysis, how we can identify the most valuable data to generate in order to prioritize our experimental resources. And then finally, I'll wrap up with a few key conclusions. All right, so introduction to agrochemical discovery and data. So as we've been hearing, well, in the agrochemical industry in general is requiring new chemicals to be developed due to emerging resistance changes in regulation. So new chemicals are required, and the most fundamental property required is that bioactivity, as George was talking about. So that's a very that's the most important property. So that's what this project focused on, was that those initial hits in bioactivity. So FMC provided some data sets across multiple, which had data for across multiple species of weed and pests. And within this data set, we also had different spray rates. So that's the amount per hectare applied. And we also had some different methods of application. These data sets were sparse and they were noisy data sets, and that noise or variability comes about largely due to the complexity of what we're measuring, and also in part due to it being a subjective measurement that's being taken. So it's a subjective measurement of that plant health. Right, so there are two data sets, one of potential insecticides and one of potential herbicides. And the insecticide data set is approximately 80,000 compounds consisting of 30 experimental endpoints. And it was 6 species of insects from the Lepidoptera and Hemiptera orders. And there were a mixture of spray rates, and it was 12.5% of filled in terms of that data matrix. The herbicide data set was quite a lot bigger. There were just over 200,000 compounds and 77 experimental endpoints. There were 15 species of plants. These were from the grasses and broadleaf type plants. Again, a mixture of spray rates. And here, for this herbicide data set, we also had a mixture of methods of application. It was a larger data set, but it was a more sparse data set. So only 7.5% of that data matrix was filled in. And there were a few data cleaning steps initially applied. So duplicate compounds were aggregated, and endpoints with low amounts of data or very low variance were removed. So where we knew there wasn't a possibility of building a good model against that endpoint, those endpoints were removed. Right, so now I'll give some background into the Alchemite method for deep learning imputation. So the Alchemite method relies on a process called imputation. Traditional QSAR methods relate structural features of a molecule called descriptors to their properties. So here we have a filled-in matrix of descriptors. So we have descriptors calculated for every compound, and we can relate those features to the experimental properties. Imputation, on the other hand, is a process which uses a sparse matrix of experimental data, and it fills in the gaps with those predictions. And it uses the descriptor matrix as well. So here we have the descriptor matrix as well as this experimental data table. And it learns from the structure activity relationships between the descriptors and the experimental data, as well as the inter-experimental endpoint correlations in order to make highly accurate predictions. But what it can also do is make predictions where there isn't any experimental data. So where for compounds which haven't been synthesized yet, also known as virtual compounds. So in these cases, it uses just that descriptor matrix and it can still make high quality predictions. One key advantage of the Alchemite methods are that it doesn't just provide a single prediction, but rather a probability distribution of values. And sometimes we see a probability distribution like what we have on the right with a very broad peak. And this indicates that the model is not very confident in that prediction. So it doesn't have enough data to support that prediction. Other times we have a sharply peaked probability distribution like what we have on the left. That indicates that the model is confident in that prediction. Also seen as a small error bar. And what I'll go on to show is that this confidence correlates very well with the observed accuracy. And so we can pick out the most these confident predictions and use those in order to guide and base our decisions. Another key advantage of Alchemite is that it generates interpretable relationships between experimental endpoints. So down on the bottom left here, we have this heat map indicating the importance of an input as a column along the bottom here in the prediction of an output as rows here. This gives us an indication of how the imputation model is forming those predictions, what information it's using. So if we take experiment A3 as an example, we can see that A2 here is the most important input in the predictions for that A3 endpoint. So we can understand the relatedness between experimental endpoints, and we can also this can help us to prioritize experiments and the ordering of experiments. Right, so how do we validate these models? So let's just clarify initially what these models are. So we have the imputation model, where we're using that sparse assay data as input in addition to the molecular descriptors, and we're filling in those gaps. Then for the virtual model, this model is built to only expect molecular descriptors as input, so it can be applied to compounds that haven't been synthesized or tested. So this is showing the build test process for the imputation model. The imputation model, we have the data matrix, we have compounds as rows, experiments as columns. Split that into an training and test sets. This is an 80-20 split. The training set then has, we calculate the descriptors for those compounds, and then a model is built using a cross-validation approach. And then we validate that model by taking each row of the test set. We calculate the descriptors for that compound, and then we'll look at each individual measured data point. We take one away. For example, here we've got this red data point. We can take the other measured data for that compound, use that to impute a value. Here we've got this purple square here, and then we can then compare that value with the measured value, and in that way validate the model. And the process for building the virtual model is very similar. again, we have the descriptor matrix as well as the experimental data matrix. The model is built using a cross-validation approach. However, importantly, the virtual model is built to not expect any experimental data as input. So at test time, all of the measured data is removed. We get the full matrix of predictions, and then we can compare these predicted values with the measured values to validate that model. So how do we show the results and assess the results? So here we've got two methods, and on the left we have a performance profile giving the overall performance of the model. We have the R-squared value, which is the coefficient of determination along the y-axis, and we've got the endpoints ranked from best performing on the left to worst performing on the right. And in terms of the R-squared, an R-squared of 1 is perfect accuracy, so that's perfect agreement between the predicted and measured values. And the R-squared value can go down to as low as -infinity, but anything below 0 is worse than random. Increasing accuracy is this line of endpoints ranked going up, so we're getting towards that top corner is going towards increasing accuracy. And the median R-squared is the metric that we use in order to show the overall accuracy of the model. Then on the right here, we have an analysis for a single endpoint. And we have RMSE on the y-axis, that's your root mean squared error. So that's showing your accuracy. And then on the x-axis is the most confidently predicted percentage of test set. And we have the average in the error calculated by Alchemite with the predictions as this, so that's that black dotted line. And then we have the accuracy in terms of how well that predicted matches up to the measured in blue. And so what we like to see is this sort of slope where we're seeing that confidence correlating with the accuracy. Right, so now on to the test set results. So let's start with the insecticide data set. We can see one of those performance profiles here on the right. So again, we've got the endpoints ranked in order from best performing endpoint to worst. And we've got the imputation model in green, insecticide virtual model in orange, and random forest in blue. So we use random forest to compare with the Alchemite methods to show us the general performance from a traditional QSAR method. We can see that the imputation method did quite a lot better. We've got a clear gap between imputation and the other models. The virtual and random forest models performed quite similarly. But we've got this clear gain using imputation. So even very sparse data has added a lot of accuracy to these predictions. So we're using that sparse experimental data and really seeing a good improvement in accuracy. Then here I'm showing the performance per endpoint. So I have these histograms, again, in imputation in green, virtual model in orange, and random forest in blue. And here we've got the Lepidoptera species, this top bar chart, and then here is Hemiptera species. So overall, we can see there's quite a lot of variation between endpoints in terms of their performance. And then we could also see that there was quite a lot of variation in terms of how much was gained using the imputation approach. So for the Lepidoptera species, there was in general a much greater gain using the imputation process. So that is, those species are gaining more from using the data from the other species. Next on to the herbicide data set. So here again, we've got the performance profile. Imputation model is in green, and we could see that there's a very large increase in the performance using that imputation model. So we're really gaining a lot of value from that data. So there's even stronger correlations between these species, and we're able to learn and make much more accurate predictions using that sparse data as part of the input. And again, the virtual and random forest models perform quite similarly. Then per endpoint, this again, and here we again see quite a lot of variation between different endpoints, and we can see that large gain in performance using the imputation model. We see that large gain for nearly all of the endpoints. And for some of these, the performance was fairly poor, and there are many reasons for this, and one of them would be the amount of data that we have for that endpoint. So what I then looked at was how the amount of data per endpoint, in terms of how much data was in the training set, that correlated with the performance. So on the left here, I have that correlation for insecticide compounds. So number of data points in the training set is along the y-axis, and the performance in terms of r² is along the x-axis. And so we can see that there was a moderate correlation for both the imputation and virtual models. But we could see that above about 15,000 data points, we weren't seeing a further gain in that performance. And this could be due to many different factors. It's perhaps that there's that data encompasses other data that is slightly a bit different. Perhaps it incorporates older data that was measured under slightly different conditions, and therefore that data has more variance. For the herbicide data set, it was quite interesting that we didn't see a positive correlation at all. We could see that for fewer than 500 data points, there was quite poor performance, but above that, there was no positive correlation. And actually, for the virtual model, there was a moderate negative correlation. That's again due to the compounds endpoints with the highest amount of data points in the training set having a lower r². So again, perhaps due to some sort of variation in that data. But what was really interesting to see was that the imputation model didn't have that same drop-off that was comparable to endpoints with less data. And so while the virtual model struggled to understand that variation, the imputation model was unable to make sense of that and make highly accurate predictions despite it. Next, I wanted to highlight quite an interesting observation that we made, which is that as we were filling in the data with these predictions, we noticed a pattern emerging. So here we have the measured data, and one of the insecticide species, the Hemiptera species 2, is an example. And we could see that as we went from the higher spray rates to the lower ones, we were seeing in general the values were getting lower. And we can see this a bit more clearly as a heat map. And if we focus in on just one of these, we can see that that is going down, and that's something that we found generally across the predictions. And when we looked at the measured data, that rate response relationship did exist there. It's not as clear cut with the measured data. Obviously, it's a very sparse matrix. There aren't many compounds that have measured values for all of the spray rates. But the model was able to pick up on that rate response relationship, understand it, and make the predictions accordingly. Right, so next on to the uncertainty analysis. Here I'll give a few examples of how we can focus in on the most confident results, and how that relates to the accuracy. So this is an example using the most accurately predicted endpoints, the herbicide data set. It was grasses species 2. So here on the left, I have one of those RMSE versus the most confidently predicted percentage in the test set. So here where we have 100% of the data, we can see that over here. And then if we remove the 50% of these predictions with the highest error bars, we then see that these data points are actually moving closer to this identity line, which is where we have perfect correlation between predicted and observed. And then if we move all but the 10% most confidently predicted data points, then we see very good accuracy. Now, I'd like to give an example where we have quite poor performance. So this is one of the insecticide endpoints. We've got an R-squared of 0.33 using the virtual model. Again, here we have all of the predicted data points. If we remove 50% of those with the highest uncertainties, we're then seeing that we're getting better in terms of that overall accuracy. And then if we focus in on just the 10% most confident, we see that those are very well predicted, that those have high accuracy. So we're seeing very good correlation between the model confidence, those error bars, and the observed accuracy. And so this allows us to focus on just those most confident predictions and use those in order to base our decisions. So even for models, endpoints, which are poorly predicted overall, we can still use those predictions in order to guide our decisions. Next, I wanted to talk about another application of Alchemite, which is to virtual compound prediction or in silico screening. So FMC supplied a virtual library of about around 1.25 million compounds curated from chemical vendor databases, and their goal was to have an activity optimized screening library that can then go into their experimental screen. So as George talked about, they wanted to have this screening library that was better, so that they could have a much better hit rate. And these compounds were predicted using the virtual model that doesn't require any experimental data. And predictions were made for all of the herbicide and insecticide endpoints. And so altogether we had about over 100 million individual predictions. And so how would we assess all of those predictions? Well, we can use Stardrop's probabilistic scoring tool for multi-parameter optimizations. We can assess multiple properties simultaneously. So for example, here we have a scoring profile across multiple species, and we can get out a score of how likely that compound is to succeed given those predictions. And very importantly, this probabilistic scoring takes into account the uncertainty in those predictions. So this goes back to looking at that correlation between uncertainty, certainty, and accuracy. So we can, in this way, assess large-scale predictions. So we can virtually screen and rank compounds on a large scale, and then focus only on those most promising. Next, importance analysis. So the imputation model allows for you to see which of the inputs, either descriptors or experimental data, which of those are the most important for the prediction of the endpoints. So here we have a heat map with the inputs along the bottom as columns, and then all of the insecticide endpoints are in rows. And the more red the square, the more important that descriptor or experimental endpoint is to the endpoint across the row. So here we've got descriptors in blue and experimental endpoints in gray, and we could see that the ones that were the most important were the experimental endpoints. So we're gaining more from using that experimental data than we are from using the descriptors. And what we could see is some boxes where related inputs are informing related outputs. So, for example, down here we can see a few boxes where we're seeing lots of red squares. So let's just focus on that area in a little bit more detail. So we could see that the L1 species were informing the L2 species. The L1 and L2 species have a very strong relationship, and the L2 species are important for L1. So we can see that kind of pairwise relationship between those, and similarly between H1 and H2. So we've got these two boxes for that pairwise relationship. And so this was really interesting to see, and really, really nice to see, because these are the sorts of relationships that we would expect to see. And so the model is doing what we would expect it to, learning from the data that we would expect it to learn from. Next, looking at the herbicide endpoints, those have been spread out across two separate heat maps. Again, we've got the inputs along the bottom and the outputs as rows. And again, we have descriptors in blue and experimental endpoints in gray, but here you can see that there are no descriptors, and that's because there weren't any descriptors as that were in the top 5 most important input for any of these experimental endpoints. So this really goes to show why we saw such a huge increase in performance using the imputation model. Where we're really gaining the most information is from that experimental data. So we had a few of these boxes where we have related outputs and informing related outputs, and mostly on the endpoints on the right. So let's focus in on that in a bit more detail. So we could see that whereas we had those pairwise relationships with the insecticide data set, we've got many more interconnected species relationships with the herbicide data set. It wasn't only within grasses being important for grasses and broadleaves being important for broadleaves, it was across these species and between those different types. And what we found was that the spray rates were very important for informing the key measure of what informed what. So we had spray rate 1 informing spray rate 1 largely. We could see that spray rate 2 largely informed spray rate 2. And this is likely due to that rate rate response relationship, so we have that on the data on a similar scale is forming the most informative correlations that the model is using. And what we also found was that the STRO method of application was informative for the STRO method of application. So again, because of it's most related to that the other data, and it learns from other species within that same application method. Next, I just wanted to mention that because there weren't these kind of pairwise relationships so much in the herbicide data, that it seemed that there were certain species that were important kind of more across the board for other species. And we could pick out certain species that were more informative overall than others. And so we picked out that G1, G2, G8, and B1 species were the most valuable experiments in terms of their ability to inform predictions in other species. So not only do you get the predictions for that endpoint, but those ones can be used to make accurate predictions of many other species or many other endpoints. So experiments using those species should be done first in order to maximize the benefit of that imputation process, and then you can reduce the time, effort, and resources in testing all compounds in those other experimental endpoints. Right, so in conclusion, the Alchemite imputation model outperformed virtual and random forest models. We saw that for the insecticide data set, we had a significant gain, and for the herbicide data set, we saw a very big gain. So we have these gains in performance even when there is very limited experimental data. And this really demonstrates the value of imputation across species. We are able to use these species correlations and that relatedness in order to make more accurate predictions. And then I demonstrated there's a strong correlation between the estimated uncertainties and the observed accuracy in the predictions. So we can identify the accurate predictions even for endpoints with poor overall model performance, and we can use those ones to guide our decisions. And then finally, I've demonstrated that the importance analysis provides key in understanding of the strength of relationship between those experimental endpoints. We can identify the most valuable experiments to accurately predict other experimental endpoints, and therefore prioritize experimental resources, thus saving time and money. So with that, I'd like to thank Matt and Tamsin, who have helped lots on this project, and also Peter Hunt and Samar Mahmood, who have also helped in terms of some scientific discussions. And then on the FMC side, George and Laurie for designing this project, and Laurie for putting together the data sets, Seva, Rob, Rachel, Eric, and Jeff as well for their involvement getting the experimental data, as well as some discussions around the project. So with that, I'll pass back to Matt.
Thank you very much, Bailey, for a really fascinating presentation, for taking us through that project and the great results. Just before we move on to the Q&A session, just wanted to go back to George and Laurie and just ask a little bit about how FMC plans to follow up on the results of this project.
Yeah, so one of the things we want to do, obviously, is to use this to inform future compound purchases. And so right now we have a set that was recently purchased and will be going into screening over the next few months. And so we're certainly very much looking forward to finding out what the activity of those compounds will be. One of the things we did in designing the set for the virtual predictions was I pulled together compounds that we had recently purchased, so prior to the start of this experiment, but not long enough ago that we had data on the compounds yet. And so those are in the early stages of screening, and so we're starting to be able to look at how the activity on those compares to the predictions that were made. And we don't have all the results yet, but the early screening results are certainly encouraging for the model's ability to discriminate and identify some of the compounds that did turn out to be active. So that's very nice to see.
Yeah, and maybe just to put a little finer point on what Laurie said there is Bailey referred to the 1.25 million compound set that we had curated from available chemistry. And so that was already somewhat optimized, but as I mentioned earlier, we can't screen 1.25 million compounds, nor can we afford to screen 1.25 million. So we selected about 10%, about 1%, excuse me, about 1%, I think, of the compounds of that 1.25 million, and they were completely selected based on the models that we got from the imputation model that Bailey talked about. So if this works out well, like we hope it does, and like the training set indicated it should, that we have a higher probability for this set of compounds of showing us some interesting biological properties, which gives us a lot more to work with in terms of future follow-up. It's it's not a real fast process. It takes us a while to go through even that many compounds, but over the course of 4 to 6 months, I think we're actually going to have, and Laurie mentioned we're already seeing a little bit of data. I think we'll have a pretty good picture of what kind of results we got at least from this first set. And we have another set that we're going to be looking at in 2023. And we actually have a third set that we're actually in the process of looking at with just in the insecticide area that are specific to one species, aphids in particular, that we're going to be following up with as well.
Fantastic. Thank you very, very much. So we're just about to move on to the Q&A session. I can already see some great questions coming in. But just before we do that, I just wanted to talk a little bit about how to actually use this method in practice. It's great to have a really fantastic scientific method, but it's also important that you can access it easily. And this is embodied by our Cerella product that uses Alchemite in the ways that Bailey has just been describing. So Cerella is really about enabling active learning in the compound optimization process. It sits on top of your data sources, so your data mart, your ELN, or even just flat files, and it automatically updates and prepares experimental data for model building. So it populates that sparse matrix that Bailey was talking about of compounds and experimental endpoints. And in doing so, it also applies all of those tedious but important rules for cleaning and transforming the data to get the best model performance. It then automatically applies Alchemite to that sparse and noisy data and updates those models as new data become available, so that you're always working with results based on the latest information and removing that burden of manually building and updating models that I'm sure Laurie will share the experience of having to do that time and time again. Of course, with the Alchemite model, you can then impute that matrix of data. So the purple squares, we fill in the blanks in that data. And that brings a whole new problem. You might have millions of compounds by thousands of experimental endpoints. If only a few % of that is actually measured in practice, that's very manageable. But if you fill in the other 90+% of that data, you suddenly got what we describe as the massive matrix challenge. You've got true big data. So Cerella scales to manage that matrix that can contain tens of billions of data points to make it very easy to surface those valuable results and conclusions from that we've just seen. And of course, making that very seamlessly available through our Stardrop software is one interface for it, but it's also possible to link to this from other platforms via standard sort of RESTful APIs. So if you have other ways to analyze and visualize that data, that's certainly just as open to that as well. And now on to the Q&A session. So let's see what we've got here. So here's a really good question. Says the virtual models seem to not perform so well on average. So probably due to the challenging nature of the data, as we've heard. Does virtual screening make sense, as the quality of predictions for a large number of endpoints would be questionable? Bailey, do you want to take that one?
Yes. So as I showed in the uncertainty analysis, where we don't have the kind of performance that we would like, it doesn't mean that we need to throw out all of those predictions. What I showed was that the uncertainty correlates very well with the accuracy. So if we pick out just the most confident predictions, we can use those in order to guide our decisions. And using something like probabilistic scoring, like I said, that takes into account that uncertainty. And so the score that you would get out of that probabilistic scoring takes into account that uncertainty, and you can see what the chance of likelihood of success is, and that's a real value, and it has that uncertainty there, and so really you don't need to get rid of all those predictions. You can pick out those ones that are accurate.
Yeah, that's a really important distinction, as Bailey says. With a conventional QSAR model that doesn't have a good uncertainty estimate, you just have to throw away the whole model. You couldn't use it. But here, a proportion of those predictions are very confident and hence very accurate. So you can still gain a lot of value in prioritizing those most likely to achieve the outcome that you're looking for. Absolutely right. Okay, so here's another good one. Have you looked at merging data from the different applications or indications together? Would you expect that to work, or at least be as good and not worse?
Right, so I believe you're asking whether we should merge the insecticide and herbicide data sets together, and whether we would expect it to be better or worse. So I think we did see very strong correlations, and perhaps even stronger correlations than we expected with a herbicide data set. But I think that between weeds and pests, there are great differences in the bioactivities. So we would expect that, because those species are so unrelated, that it wouldn't be able to gain, or any gain that it might make might be a bit artificial. So we felt it best to keep them separate. So
yeah, that's absolutely the case. We kept them separate. However, what I would say, based on our experience in other projects, that merging them together would not cause a decrease in performance. Whether you'd get any increase would depend on that strength of correlation, and also depend on how many compounds had data from both indications or both applications. But one of the really powerful things about this Alchemite method is that you can merge compounds and data for different indications with different endpoints. And unlike many multi-target modeling methods, we do not see a decrease in the performance, even if these indications are completely uncorrelated. We did a really nice example with the University of Dundee, where we looked at making predictions with a global model across different global health indications like TB and malaria, schistosomiasis, and so on, merging that data set from many different projects, and we're able to show no drop in accuracy at all. So yes, you can put all that data together and you'll get at least as good, if not better, performance. Another question here. This one I think is probably best targeted at Laurie, if that's okay. So we had this 1.25 million compounds for the virtual screening. Can you say a few words about how they were curated from the very long list of available compounds?
Yeah, and that got to be a somewhat complicated process that I think all of us were having some discussion about for a while there, because a lot of this curation took place right around the time, unfortunately, that the war in Ukraine broke out. And so who you could buy compounds from and what were the best sources was very much in flux for a while. And that informed what we ended up doing. So there are actually a couple of blocks of chemistry here. As I mentioned, one is compounds that we have in-house, but which don't have activity or which don't have data for some or all of the endpoints. And so that's useful. That was basically a sure thing that we could get it if we needed to. And then ultimately, Enamine was happily able to bring their labs back online and start selling again. And so that was the bulk of what we bought. And then we also supplemented with a couple of pretty loosely curated sets from some of the Synthesis on Demand collections that were mostly there as a stopgap in case we couldn't get anything else. But the main body we worked with was enamine off-the-shelf chemistry, which had been processed through some of our usual internal criteria for compounds that we would consider purchasing. And then, as we've already discussed, the models were the main input into deciding among those compounds which ones we actually bought.
Thank you very much, Laurie. I think we've got time just for a couple quick ones. So here we say, based on the MPO profile built on the multiple models for targeting various targets, the insects or weeds, do you expect a drop of selectivity for the selected compounds for purchase? Bailey.
Sure. So within this data set, we were looking for just activity across multiple species. We weren't looking at selectivity in particular. However, you can set up a multi-parameter optimized scoring profile looking at selectivity. You can simply set the criteria for the ones you want it to be active as active, and the off targets as inactive, and it will work in the same way. Rank the compounds based on that selectivity that you want.
And I think one more, and again, apologies to everyone who we can't answer all the questions today. We will follow up by email, but the last one I'll just mention is what type of structural information did you feed into the models and how did you do it? So I guess this is relating to the descriptors that we used.
Yes, so we used our standard descriptors, startup descriptors, which we've tested many times and found them to be very, very good descriptors, which very often outperform or at least as good as other structural descriptors that we've come across.
Yeah, so they include sort of whole molecule properties like usual things, molecular weight, log P, McGowan's volume, charge, flexibility, and so on, but also about 300 2D smart space descriptors capturing key structural features and their relationships to one another. So, you know, nothing magical, but a good, solid, widely applicable set of descriptors that we've used many times before. And so finally, just to draw a close then, again, to say a big thank you to everyone who's joined us today, particularly those of you who have stayed on the line past the hour. And indeed, a big thank you to George and Laurie, first of all, for collaborating with us on this project, but also for joining us today and providing that sort of great insight into the motivations and results from the project and answering some of the questions as well. So thank you all very much, and we hope you'll join us for our new webinar series early in the new year. Thank you very much.